Hello guys, and welcome to our first sample examination, Kenya Certificate of Secondary Education, Paper 1, Biology. We're going to do it from question 1 to the last. I hope you're going to understand, you're going to get something by the end of the revision. Please remember to follow on Twitter. All the accounts are showing down there. So let us begin. So we are doing the first sample examination in Certificate of Secondary Education, Paper 1, Theory paper. This comprises of 80 marks. So let us start with the first one. The first one says the figure below is a diagram of a skull. So use the diagram to answer the questions that follow. So that is the skull that we have. That is the skull. Look at the skull. And therefore, let us move the question. The first question. Identify the type of food utilized by an organism in such a skull. This one is only 1 mark. Being asked to identify the type of food utilized by an organism in such a skull. Now, we're going to give a reason later, but looking at the skull, it feeds on grass, vegetation. And the reason for this, they're asking, give a reason for your answer in A above. The reason is you look at the arrangement of teeth, we look at the dentition of an organism, we look at how the teeth are arranged, and therefore from the dentition we can be able to deduce and be able to say what kind of food that organism feeds on. And therefore, looking at what we have here, you look at this one, you see it has, when you look at the upper jaw, you discover that at the end of the upper jaw, towards the posterior end of the upper jaw, discover that we don't have teeth. There's lack of teeth. And therefore, this one definitely shows you that this one is an organism which feeds on grass. A good example is the likes of goats, cows. And therefore, we're saying the absence of the upper incisors indicating presence of a horny pad against which grass is pressed for cutting. And also number 2, you look at that, there's a gap there. That gap between the canines and premolars, we call it the diastema, for manipulation of food, and also the turning of the tongue. Such kind of organisms have a very long tongue, therefore there's that issue of turning, so it must have a space for that. Then question number 2, explain why specimens are important in studying biology. Why do we need specimens in studying biology? And this one is a very simple, that is a proper study, for a proper study understanding of living things in the laboratory. A specimen is just a sample of the whole population. We take a sample of the whole population. A specimen represents the other organism in the population. Therefore, this one will give us a proper study and understanding of the living things in the laboratory. Those living things which are outside the laboratory. Also, another question, identify an apparatus that is effective in capturing a scorpion, for example. Now we know a scorpion, we know what a scorpion is. And here, remember, a scorpion has poison gloves. So we use a pair of forceps. Remember, it's very poisonous, so a pair of forceps will work. Butterfly, very highly mobile animals like butterfly. We use a sweep net, you sweep them around. Next, identify an organelle that is most abundant in proximal convoluted tubule. The word abundant here means in plenty, in the proximal convoluted tubule. And we know the proximal convoluted tubule is part of the nephron. And here we talk about the mitochondria, or what we call mitochondria. Why? Because this is the part where energy is highly required. Give a reason for your answer and here is the reason, is because yield energy, it provides energy or yields energy for the active transport in the proximal convoluted tubule. Then there's a question here. A student enclosed a leaf shoot using a transparent polythene bag. After 45 minutes, clear droplets were observed on the walls of the polythene bag. Anhydrous cobalt-2 chloride paper was used to determine the identity of the clear droplets. So the first question, identify the process under investigation. This is a transpiration process. This is transpiration, and it can be deduced by the use of polythene, and also what they were talking about, the clear droplets. The clear droplets here are just water droplets. State the expected color changes of anhydrous cobalt-2 chloride paper under the same. So because we know that the droplets are actually water, so anhydrous cobalt-2 chloride paper shall be it will turn from blue to pink. Then explain why the clear droplets cannot be observed when the leaf shoot is not enclosed in a polythene paper bag. And this is because plants lose water in form of water vapor, and you cannot see water vapor. Water vapor is like air, so you cannot be able to see it. And the figure below illustrates a relationship between developing fetus and the maternal tissue. So this is what we have. There's a developing fetus, and then that is, we are assuming that is the maternal, inside the maternal tissue. There's part L, then there's part K, which I think we're supposed to use them in the answering of the question that follows. Therefore, the first question they're asking, give 2 rows of part labeled K. First of all, we must know what are these part labeled K. Part labeled K is simply a placenta. So question comes, what is the function of the placenta? And this one offers exchange of substances between the mother and the fetus. Then number 2, it also does the production of hormones. It's the major one that produces hormones, so that the hormone that maintains the pregnancy. Name a vessel in L that supplies nutrients and oxygen to the fetus. Now, L is actually an umbilical cord. So we are looking at the vessel in L that supplies nutrients and oxygen to the fetus, and this is just an umbilical vein from the word umbilical. Then explain why deficiency of vitamin K leads to excessive bleeding even in small cuts. Explain why deficiency of vitamin K leads to excessive bleeding even in small cuts. Now, vitamin K, we all know, vitamin K is required for synthesis of prothrombin. Now, prothrombin is converted to thrombin that activates fibrinogen to fibrin. We all know what fibrin is. Fibrin is that wire, that mesh that prevents the excessive bleeding. Identify the parts of the mammalian heart that plays the following roles. So role number 1, prevents oxygenated blood from mixing with deoxygenated blood. Preventing oxygenated blood from mixing with deoxygenated blood. And this is the septum. The septum is in between the heart. Between the 2 chambers of the heart is a septum. That septum is the one which prevents the mixing of blood. Number 2, the part which secretes pericardial fluid, that is the pericardium. Prevents valve from turning inside out. Those are tendons. Tendons in the heart prevents the valve from turning inside out. Now, a flower was observed to be scented. Identify the phylum of living organisms that transfer pollen grains within the species of the plants having the flower. So it was scented, and therefore these are arthropoda, because we are talking about the phylum. Because we're talking about insects, most of the insects are the one which transport, which does the pollination, but the insects are under class Insecta, phylum Arthropoda. So if you have been asked the phylum, then it is the phylum Arthropoda. Name a physiological process that aids the organism in A above affect the transfer of pollen grains while giving a reason. We're talking about diffusion here. That is, send diffuses from flower region, which is highly concentrated, to the region of low concentration. Explain why maize stalks do not undergo secondary growth, yet they increase in girth, but they never undergo secondary growth. Maize are monocots. They have no vascular cambium, which is responsible for secondary growth. So increase in girth is due to enlargement of primary cells. So that's why they don't undergo secondary growth. A food sample was made into suspension, and few drops of iodine was added into it. A blue-black color was obtained. A Benedict's test on the sample gave a clear blue-green blue color. Another sample of the same food was made into the suspension. Some millet seeds which had been soaked for 48 hours were crushed and mixed with the substance. The mixture was incubated in warm water bath for 6 hours at 35 degrees Celsius. After incubation, the sample gave a negative test with iodine and a positive test with Benedict's solution. So, question number 1. Explain why the iodine test was negative after incubation. The answer is very simple. Starch was hydrolyzed to reducing sugars by enzyme present in the germinating seeds. The germinating seeds have enzymes, and therefore they act as catalysts. So they will catalyze, and therefore there will be a hydrolyzation of starch to reducing sugars. So with a test of iodine, remember iodine test is testing for starch, which has already been hydrolyzed to reducing sugars. Explain why it is necessary to incubate the mixture in warm water bath. And this one is because of the optimum temperature, that 35 is optimum temperature. Explain why rate of absorption of mineral salts reduced with less oxygen concentration. Explain why rate of absorption of mineral salts reduced with less concentration, with less oxygen concentration. Now, anytime you hear the word concentration of oxygen, it means the process requires energy. And if the process requires energy, then with a reduction of oxygen, because oxygen is the one which oxidizes food to produce energy under respiration, then it means even the energy production will be low, and therefore the reaction rates also will go down. Oxygen is required in the process of respiration to yield energy for active transport. I want you to name the following: the material that strengthens xylem tissue. The material that strengthens xylem tissue, we all know them. Those materials are lignin. Then the tissue that's removed when the bark of a dicotyledonous plant is ringed. That one is a phloem tissue. Then stage 2 function of a cell sap. Function number 1, it maintains the cell shape. Then function number 2, it contains sugars and salts, hence cause what we call osmotic pressure. Those are the function of cell sap. Then below is apparatus used to estimate the population of an organism, especially plants. That is the apparatus, and we all know this one. We all know this apparatus. This one is it's a quadrat. Now below is an apparatus used to estimate the population of an organism, especially plants. That is the apparatus, and we all know this apparatus. This one is the quadrat. So the first question, what is the name of the apparatus? What is the name of the apparatus? This one is a quadrat. Then explain why the apparatus is not suitable for estimation of animal population. Now remember, the mechanism of using this quadrat is by throwing. We will do it by throwing, and animals are highly mobile. So, and then they'll also be running for their lives. So if you try to throw the quadrat whereas an animals will tend to run away. So you cannot be able to. And then you are supposed to count organisms within the quadrat. So it may be quite difficult because animals are highly mobile. Now state one feature that can be used to identify plants under enumeration above. State one feature that can be used to identify plants under enumeration above. Any feature. This we can look at flowers. For example. Below is a photograph of plant taken at different times of the day. 6 a.m. the plant looks like that, then 12 p.m. the plant looks like that. So let us look at our first question. Give a term used to describe the nature of cells obtained from the plant at 6 a.m. The cells are turgid. That's why the plant looks like that. Then at 12, it is what? Flaccid. Now, what is the significance of this phenomena to herbaceous plants? The phenomena of turgidity and flaccidness. So this one gives support to the plant, that is thus exposing the leaves for photosynthesis. And the figure below illustrates the blood supply and drainage of the liver. That is the figure that is showing how blood is supplied and drained from the liver. Now you can see the heart there. You can see the descending aorta. We can see the heart there. You can see the descending aorta. You can see the liver. You can see the intestine. You can see the inferior vena cava, liver. Therefore, first question, in which of the vessels labeled 1, 2, and 3 would you expect highest concentration of glucose after an overnight of fast? And then you need to give a reason. Now, this is part 3. Because part 3 contains what we call the stored glucose, glycogen, which is broken down to glucose due to low glucose concentration by hormone glucagon. The illustration below represents the base sequence of a segment of a nucleic acid. That's how it looks like. G, A, C, A, G, U, A, C. G stands for guanine, F adenine, C cytosine, and U for uracil. So, first question. Which nucleic acid does the above segment represent? And this is because of U, this one is messenger RNA, what you call the RNA or the messenger RNA. What is the reason? Presence of uracil. Name the part of cell where the segment of nucleic acid is used. Ribosome or cytoplasm. Describe how the following structures lower the rate of transpiration. Sunken stomata. How the sunken stomata? Water vapor accumulates in the pits, lowering saturation deficit. Hair leaves trap a layer of moisture, lowering saturation deficit. Now explain the following observation made by Gregor Mendel's experiment. A cross between a pure breed tall plant and a pure breed dwarf plant had all the first filial generation being tall, 2 marks. This is what we call complete dominance. Gene for tallness was dominant. Now, during an ecological trip, student came across a bone. During an ecological field trip, student came across a pond with several bird species. A Upon closer look, the student noted that the beaks of the birds were different in length, shape, and size. Identify the type of evolution responsible for the beak's length, shape, and size. Divergent evolution. What is the significance of phenomena to the birds? This one allows organisms to explore different ecological niches and eliminating competition. Now, explain why excessive use of pesticides in the soil lowers the concentration of nitrates. Pesticides kill nitrogen-fixing bacteria that convert atmospheric nitrogen to nitrites. Explain why a total of 1,000 animals that include giraffes, elephants, and zebras can occupy 10 acres of land without depletion of food, and yet 1,000 cattle will deplete resources in the same piece of land. Because when they explore different ecological niche, feed on different parts of vegetation, unlike cattle. Name 2 end products of anaerobic respiration in animals. And these are energy, lactic acid, or carbon dioxide. Explain why the presence of air bubble in xylem vessel will cause drying up of a plant. This is because it breaks the cohesion force between water molecules, interrupting the continuous flow of column water. Identify a useful excretory plant product obtained from raw pawpaw fruit, that is papain. Give the use of the plant excretory product name in one above as a meat tenderizer. Name the method by which the following excretory products are removed from plants: excess water, by transpiration, carbon dioxide,